Hello, and welcome to our presentation on gaslight gatekeep garbage, raccoon foraging, and human cues. For our project, we decided we wanted to look at how the rise of urbanization and thus deforestation of a lot of species' natural habitats can lead to an increase in urban wildlife as these now homeless animal populations learn to live in a new urban environment and how these different populations of animals affect each other. So we decided that our main question would focus on raccoons as they are common urban wildlife that have adapted very well to this new setting. And we specifically decided on the question on does the presence of people affect raccoon feeding behaviors? We also took a little look at other factors such as temperature, location, and time in case they also played a factor in affecting raccoon feeding behaviors. The method in which we decided to approach this was to study the number of humans passing through an area and if that affected the number of raccoons entering an area to feed by observing the raccoons' behaviors in a couple of different feeding locations. For our prediction, we thought that as human foot traffic increased, the number of raccoons present would also decrease.
To investigate whether raccoons' foraging behavior is affected by population density, we first outlined the areas where populations of raccoons are known to forage and have consistently been cited by locals around campus. We then collectively divide the experimental sites and camped at each site for up to 1 hour during a 4-hour time block. Specifically, this is 8 p.m. to 12 p.m. Because the species is relatively nocturnal, all observations were made an hour after sunset. Observational sites were then limited to campus perimeters at the University of Texas at Austin, and we ended up narrowing it further down into 4 sites: the Norman Hackerman Building, William C. Power, Brackridge Pathers Dormitory, and Cypress Bend Cafe. Once we identified our sites of interest, we then repeated this procedure. Using a manual tally counter, we then incrementally counted the number of pedestrians, including individuals passing by, and the number of raccoons present during that time point over the course of 3 weeks. The temperature and the time of the evening in which the data point was collected were additionally taken as an independent variable. All data points were then imported into a statistical computing program for further analysis.
Lead us not into temptation, but deliver us.
And we conducted a correlation test and a Kruskal-Wallis test.
Moving on to our results section, as Angela just discussed, we performed a linear regression analysis on our 2 main variables, the first being the number of raccoons that visited the area during the observation period, the second being the number of humans that passed through the area during the observation period, and we ultimately found no significant relationship between either variable, as evidenced first by a p-value of about 0.95, which is very non-significant, as well as this little scatter plot here. So you can see the line of best fit is pretty much a straight line, and the R-squared value, which looks at the strength of the correlation, is pretty much 0, which essentially means no correlation.
Why are we still here? Just to suffer?
And so we also wanted to evaluate the effects of confounding variables upon the visitation of raccoons, and we specifically did a linear regression analysis for temperature, and then 2 separate Kruskal-Wallis tests for location and time of evening. The Kruskal-Wallis test basically being an ANOVA, not to get too far into the details. And we ultimately found no significant differences with these variables either. And this is just kind of evidenced in this little box plot here, where on the x-axis you see the locations that we recorded in, and the y-axis is the number of raccoons. And you can see there's not really a huge difference between any of the values for each different location. So the big takeaway for this study is that ultimately we couldn't find anything super significant for any of our variables necessarily.
And after collecting and analyzing our data, we found that they had no correlation. In short, this means that a raccoon should not be deterred or inspired to get food when humans are present. We hypothesized that human foot traffic would cause raccoons to avoid specific areas due to fear of confrontation, but our results are contrary to our inference. These results are significant because they can still be applied to better understand the relationship that humans have with animals in their shared environment. For example, if a city is debating on tearing down a row of abandoned houses with a large surrounding raccoon population to build an outdoor mall, but is concerned about affecting the raccoons, they will be able to use our data to make a well-informed decision based on the fact that human presence does not disrupt raccoon foraging behaviors. Our functional sample size is lower due to us having so many zero values, and this could explain our lack of any statistically significant results. Unfortunately, this means that any significant differences that actually exist are outside of the capabilities of our current data. As for limits, our experiment has one limitation that greatly influenced our results. We were only able to study raccoons on a college campus. Much like a popular national park, these conditions can create animals that are socialized and well adapted to human presence. Both environments have a constant supply of food from humans, heavy foot traffic, and people seeking out interactions with the animals. We saw multiple instances of people baiting the raccoons and chasing them to get a photo. In addition, a college campus will have people present throughout most of the night. Socialized raccoons don't care about people. Their mind is set on the box of canes in the trash. To correct this, we would repeat our experiment in environments that vary in levels of human interaction, like an urban sprawl or the suburbs or a forest, etc. This will create data that has less socialized raccoons and therefore eliminate our limitation and provide more accurate results. Most people have a total disregard for the ways in which their actions affect animals around them, but in doing so, there's a possibility of higher death rates for those populations, and in extreme cases, extinction. We think that the way in which we view our relationship to the animals around us should be similar to the way in which we define health for our own species. The World Health Organization defines health as a state of complete physical, mental, and social well-being, and not merely the absence of disease or infirmity. Obviously, we are not suggesting that we give raccoons therapy or make sure that they're hanging out with their friends, but we do want to emphasize that health isn't merely an absence of a threat. We believe that it's necessary to understand the behavior and responses of animals to our actions in order to not only protect both parties, but have them thrive as well. This is a key factor in helping sustain the environment that we live in. All species must be protected, no matter how common, to avoid the threat of extinction. We studied a common animal and found no correlation between its behavior and a specific human action. However, this is still useful information because we can apply what we have learned to find ways animals and humans can cohabitate and further the understanding of animal behavior as a whole.